Hi everyone, Tonga Sua, welcome. I think this is our first AMS Anglo-Malagasy meeting of the year, just as the clocks are about to change, so it feels like spring is in the air. We're delighted to be joined by Dr Megan Aylward, who hails from the Bristol Zoological Society for their Madagascar conservation project, and she's the manager there. And we're going to learn a lot of things that have been happening in the northwest of the island, specifically around the ii, as well as wider conservation monitoring that's going on in the area, and how all of the remote DNA sampling and method of genomics has helped understand the lemur distribution, the threats due to the ongoing and more recent fragmentation that's been happening in Madagascar of the forest itself.
Great, thanks, Chris. So yeah, I just want to start by thanking Dan for inviting me and Chris for hosting today. So I'm going to talk a little bit about ii population genetics. This is a project that I was working on during my PhD, so a little while back. It's been really nice to kind of have the opportunity to think back to some of this work. And just refresh a little bit about kind of where we are now with ii population genetics, what we know and where the gaps still are. Yeah, as Chris said, I am the project manager for Madagascar Conservation Project at Bristol Zoological Society. So I've been there since about May, joined the team there who are working on the programme in northwest Madagascar. And today I'm going to talk a little bit about my PhD work, which was looking at ii phylogeography. I think we have around 30 minutes for my talk today. So then we have a little bit of time at the end. I'll also discuss kind of the general work that we're doing in northwest Madagascar as well. And so before I begin, just to mention during my PhD work, that was based at the University of Calgary in Canada. So working with Steg Johnson there, but also working alongside Madagascar Biodiversity Partnership, and a lot of the genetics kind of lab work and bioinformatics work was done at Penn State University there. So it was kind of a really nice collaboration among all of us to get this work off the ground. So I'm going to kick off and show you guys a video. I think a lot of people are familiar with iis, but this is a video I've seen many times and never get bored of, and it just really highlights their very unique adaptations and foraging. Okay, so that was just the quick video there. And just to highlight, iis do have this really unique adaptation, and their foraging behavior is largely kind of centered on this ability to locate and extract insect larvae from within tree trunks. And so we know that when iis feed on trees, what they're doing is tapping with that middle finger, listening, and then digging out any insect larvae inside. And that forms quite a large part of their diet. So when you walk through the forest, often you see these kind of traces on some of the trees in the forest, but also they eat some fruits and nectar and other things. And one of the seeds that they feed on is the rami or canarium seed. So they kind of chew into that as well, and then use their finger to dig out the endocarp there, which is rich in protein for them. So they have these incredibly kind of specialized adaptations, which actually allow them to be generalists across the island. So it means they really are able to survive and persist in a whole variety of habitats, just because they're able to eat these different food sources that do occur across all the habitat types across a lot of Madagascar. And so this map here is really just showing their distribution. So it's all along that east coast, that kind of rainforest habitat, way up into the north of the island where it's much warmer, and then also on the west coast here, so where it's kind of dry deciduous forest as well. So they've got probably the most extensive range of any lemur species that we see on the island. It just doesn't cover the kind of central highlands or the southwest of Madagascar. But aside from that, they do occur in a lot of habitat. And behavioural data that was collected has shown that male aye-ayes can have home ranges of up to 10 square kilometres. So that's kind of like during a year of their life, in their day-to-day activities, they travel these huge distances. So it's kind of their feeding behaviour plus their ability to travel these long distances could explain kind of why we see such a broad distribution across the island. A lot of work's been done, or not a lot, but some work has been done kind of since the late 80s by Eleanor Sterling, who kind of found out a lot of the early information on aye-ayes. So she really looks at some of their behaviour on Nosy Bay in the northeast of the island. And then more recently, aye-ayes have been radio collared and studied a lot more in the east of the island by Madagascar Biodiversity Partnership and the graduate students like Nico and Tim, who have spent a lot of time looking at aye-aye behaviours. And so we're starting to get a bigger picture of kind of understanding aye-ayes, understanding how they move, how they disperse, and the different things that they do. And in terms of their genetics, we also know a bit of information. So PJ Perry did some work back in kind of 2011, 2013, and sequenced the genome of aye-ayes. And we know that they have very low levels of diversity, which could be related to the fact that they occur at very low densities and, you know, are spread quite far across the island. So they're a bit more homogeneous. They're able to move between different populations. And he also sampled these populations in the north of the island and showed that even though they're relatively close together, they really do split into kind of three distinct genetic clusters. When we look at how they may cluster together, when we split three populations, they don't really share much genetic material among them. When we look kind of, if we only broke it down as two populations, then it seems the northwest and northeast are a bit more similar than the one way up in the north. And so we're starting to get a bit of an idea of how aye-ayes spread across the island and their genetic diversity. But prior to starting my PhD project, I was talking to Ed Lewis and Steg kind of about how do we sample across the rest of the island? You know, aye-ayes are very cryptic, they crawl through the forest at night, they're solitary. Even if you can find them, they're very difficult to sample. So that had us starting to think about using a different method to try and sample some of these other areas on the island, because we know in the north, those three populations are really distinct, but they are separated in some ways by, you know, a really high mountainous area, the Saratana Massif there, and there's some of these rivers intersecting that area as well. And so the idea was to try and sample more aye-aye populations across the island to try and get a better picture of aye-aye population kind of differences or similarities. And this can really kind of help us to understand some of the biogeographic hypotheses for the island. So we know, like, people are still a little bit unsure about why we have all of the different lemur species on Madagascar that we do. And we know we've got this like incredible radiation there, but really trying to understand like what barriers there were or what kind of facilitated movement of lemurs across the landscape can really help to understand kind of what's led to the high endemism that we see on the island. And so for my PhD work, the idea was to really use these samples that are already collected in the north of the island, and then go to two other sites in the island. So we have the Singi de Bianca in the west, which is this dry deciduous forest. Very little work has been done there on aye-ayes in the past. It's you know, it's quite out of the way, a bit less accessible than other places. And then Manumbu as well, which is down in the southeast of the island. And this is in the eastern rainforest, but further south than some of the populations that have started to be looked at. We know that Madagascar Biodiversity Partnership have got some work going on kind of in Kanzavatu and Tora Futsi on the east coast. So the aim with this method was to really see if we could sample populations where people aren't really working at the moment on IIs to see whether it would be a feasible method to just kind of turn up to different areas and be able to sample the feeding traces and get DNA. And so what we did was essentially at each of these different field sites, just walk through the forest and look for feeding traces. And the nice thing about looking for the traces is that we didn't have to work at night when the IIs are active, but rather we could just walk along transects in the daytime and look for some of these really distinctive traces that are found on trees. So typically when you start to see feeding traces on trees, you can see some like tooth marks on the traces. You start to see kind of gnaw marks, and sometimes this kind of hexagonal shape as well, where they've kind of tried to gnaw in all the different sides of the tree in it to enable them to access the food. And so when we found the traces, what I would do is use essentially just a sterile scalpel blade and cut very small pieces of wood from around the edge of the feeding trace. So typically anywhere where it looked like the II's mouth or buccal cavity had kind of come into contact with the tree. And so just taking small shavings of the wood from these sites and then storing them just in small tubes until we could take them back to the lab to be processed. And so I won't go into too many details here, as I know we're quite a broad audience of expertise, and not everyone's biologist, but essentially we extracted the DNA in the lab and then used a method called target capture that really allowed us to draw the II DNA away from any other DNA that was in the sample. So there would have been, you know, like wood and bacteria and fungal DNA in there as well. But we used a method to just get the II DNA kind of away from the other DNA. So we used this method in the west, in Bianca, and far in the southeast from Manumbu. And we were able to get mitochondrial DNA from that. So mitochondrial DNA is DNA that's inherited from the mother and has many, many more copies than your typical nuclear DNA. So we were able to get kind of something. It doesn't quite give us the full picture of what's going on, but it still allows us to get some DNA from these samples. And then we combined the data from these samples with fecal samples, so scat samples that have been collected in the eastern rainforest populations, and then also included some blood samples that Madagascar Biodiversity Partnership had collected kind of over 10 years of working across the island. So they'd opportunistically sampled some blood from individuals in the north and on the east. They'd also collected some fecal samples, and then we kind of were using essentially everything we could get to kind of sample across the island to try and build up a picture of how different these populations were. And so this diagram here is just kind of showing the differences between populations, and each branch is essentially when populations were splitting or diverging from one another. And what this showed us when we were looking at the mitochondrial DNA was that the northernmost population, the one way up in the far north of the island, probably just diverged earlier than all of the other populations. So that would have diverged, and we imagine there would have been kind of dispersal to these other areas as well. And then after that, the western population split off, and then the southeast. And then when we look at the kind of east, northeast, and central east, these kind of had movement and gene flow among them until much more recently. So it does suggest that within this area, there probably was movement of individuals, and it wasn't as restricted, whereas the north, almost like a few 100,000 years ago, split off from the other. So one thing that people have discussed when looking at lemurs, not just aye-ayes, but lemur populations in Madagascar, is whether populations may have split kind of from north down to the south of the island, or if there would have been a east to west split across the island, because we've got the central highlands now, which don't contain much forest. And so you don't have to worry about this table too much, but essentially what we're looking at here is just pairwise comparisons of how similar each of the populations are. And so when we look at the west population, this one over on the west coast here, compared to the other populations, it has the lowest level of divergence with the eastern population. So a higher number basically means the populations are more different. And what we see with that population is that it's more similar, the eastern and northeast population, than it is to the northwest population. So this was quite interesting for us. It really does show that the west population is probably more similar to those in the central east, which suggests there may have been more movement across that part of the island than there would have been from north to south here. And that's a pattern that we've started to see in some other lemur species as well. I think in mouse lemurs, there's been some genomics work that also shows that there's this kind of east to west relationship rather than a north to south relationship, and suggests that at times the west coast and the east coast may have been connected through kind of riparian vegetation. So that's the vegetation that stays around some of the large rivers that intersect the island there. Okay, so just a break for 5 seconds. I know that's kind of getting a bit more technical, talking about the DNA and genetics. So we've got one more slide that is technical, and then I'll move on to kind of summarizing and talking about some of the conservation work. So lastly, when we looked just at the nuclear DNA, we didn't have nuclear DNA from the west coast, from the eDNA, or from the southeast. But it allowed us to compare some of these populations. And so this chart is really just showing the proportion of genetic material shared among those populations. And so kind of to reflect earlier work, we still see that split of these 3 populations in the north, although a couple of individuals from the northwest population do share genetic material with the far north and one with the east. And then in the southeast as well, we see that that is quite a distinct population. You know, it kind of doesn't really share as much genetic material with other populations. And then on the east coast here, it's a little bit more mixed, and maybe that's what we'd expect with it occurring in the middle here. However, we're seeing that it probably shares more genetic material with those individuals up in the north of the island, and a little bit with those down towards the southeast. But this does suggest that, again, these kind of 3 populations, the northeast and northwest and eastern population, may have been connected more recently than the other populations. And so this fits in with the hypothesis that suggests that there could have been movement through riparian vegetation when forest cover kind of contracted over time. Just to summarize that, essentially what we're showing is that mitochondrial DNA indicates that there was east to west gene flow among populations, which is quite exciting because it suggests that there could have been forest connectivity across the island in the past. The furthest northmost population diverged first, so that is most distinct from a lot of the other populations. And there is some support to suggest that riparian vegetation during kind of the Pleistocene glaciations may have played a role in the patterns of genetic diversity that we see across the island. But aye-ayes aren't completely restricted to those predicted boundaries there. So they do kind of traverse some of those boundaries. So in summary, we know that aye-ayes are really widespread, but they're very challenging to find and sample in the wild. Relatively little is known about their genetics as methods improve. We're starting to learn more. And we now have this method where we can actually get DNA from the gnaw marks of eyes. So that's a really great way for people to be able to just go out into the forest. It's kind of low tech method to be able to just sample the DNA from these different feeding traces. And we do see some support of east to west gene flow. So now that's kind of that genetic part of my talk in a nutshell. I'm going to switch gears and just give a little bit of an overview about Bristol Zoo's conservation work in the northwest of Madagascar. So kind of switching from eyes to these blue-eyed black lemurs that are found on the northwest of the island on that one peninsula. And so Bristol Zoo have been working in Anglo-Malagasy for about 10 years. Previously, there was a lot of lemur behavioral ecology work that was done in this area, some amphibian surveys as well. And it's really only been in the last few years that we've kind of really switched focus to actually think about conservation and how to try and protect some of the species that we're interested in up there. And that definitely was the driver for me to switch from kind of this core research to working with Bristol Zoo, where we can actually start to think about how we're going to save some of the species that are found on the island that are likely to disappear if we don't give them much attention. So Anglo-Malagasy is kind of similar to much of Madagascar in the fact that it has a lot of subsistence farming. Habitat for lemurs is very fragmented. And on this peninsula, there's kind of two areas of forest where we focus a lot of our work. So we have Anglo-Malagasy, and that's where our field station is. And there's a few villages and forest fragments around that. And then Anglo-Malagasy in the kind of northeast of that part of the peninsula, which is a much more like continuous and intact forest, but it still suffers from some degradation and kind of small scale mining in that area as well. And so in terms of habitat restoration, a lot of work was done recently by Bristol Zoological Society to try and understand how to get trees to grow better in the region. So it's much more difficult in the west of the island to kind of get trees growing fast. Like they grow much slower than they do in the east in the rainforest. Just because the climate is a bit more arid, and I think a lot of the trees are adapted to grow slowly. So use some kind of experimental plant, reforestation plots, and looked at some different methods of how to cultivate the saplings, how to plant these out into the field, and see which ones really were more effective. And so we kind of changed our method. Initially, we were just digging a hole, putting this sapling in, and then putting soil around it. Whereas now we've gone for kind of this double pitted method, so to speak. So we dig the main planting hole, then dig another hole further down, put the sapling in there with a mix of dung and sand to allow for kind of more nutrients, but also some drainage. And then it's somewhat sheltered from the sun by this pit that's above it, and it allows for pooling of water a little bit during the rainy season as well. And then gradually that fills in as the tree grows, as the soil kind of gets washed into that hole. So we found that's actually much better for saplings survival once we plant them into the ground. And then kind of the other streams of our conservation work are community engagement and biodiversity monitoring. So our community engagement is really trying to understand what people's livelihoods are like in this region. So we have a bit of an idea, but we're trying to really bring together like what people's needs are, what could change potentially to reduce kind of reliance on some of the forest resources, and whether there are any ways to mitigate some of the threats to the species that are found on the island. And then we also have biodiversity monitoring. So we know a little bit about the biodiversity in the area, but really we're trying to get some baseline data for some of the insects, lemurs, amphibians, plants, different things like that that are in the area, understand the phenology, so that can help us with collection of seedlings or seeds for reforestation. And we know that the tree species are a really important source for lemurs, so we can get a better idea of what to plant and when. And really our aim with the long-term monitoring is to really see what impact our conservation initiatives are having over the long term. So we're kind of coming at it from both sides. We know that there's degradation of the forest. We also know that we're trying to restore some forest. So really trying to understand how any of these changes impact the biodiversity in the region. There. So I will wrap up there. So thank you all for listening, and I'm happy to take any questions if anyone has.